Hello, this is class Bio 225, Genetics from Genes to Genomes. We are going to continue chapter 7 today. We will cover part 3. And this is Dr. Lucy Abgaryan. During last classes, we have talked about some mechanisms of mutagenesis. Today we will talk about some mechanisms that cells apply or use in order to reverse those mutagens. It is interesting that some of these mechanisms that create mutations, they also trigger a mechanism within the cell that is going to reverse that mutations. Let's discuss some of them now. So we talked that there are environmental mechanisms, there are chemical mechanisms that are causing mutagenesis, right? So one of the chemical agents that was causing mutagenesis was the alkyliding agent, which was changing guanine to ethyl guanine, where it was adding ethyl group to the guanine, and it was causing GC to AT mutation. However, interestingly, this agent is also triggering a mechanism in cell that will reverse this mutagenesis. So it is also causing alkyl transferase to remove the alkyl group from the molecule, which will reduce the chance of mutation within our sequence. Another case of mutagenesis that we have talked about was due to environmental factor, was due to UV radiation, and we were seeing creation of thymine dimers post UV radiation. However, there is a mechanism in cells that can reverse it, and it's done by help of photolysis. And the interesting thing is that the photolysis is being activated by UV radiation, the same UV radiation that is causing the mutation. So when we talk about repair of damaged bases, homology-dependent repair, and it is important to remember that damage does not mean mutated genetic information. Then the repair, there are two classes of repair mechanisms here: the base excision repair and nucleotide-based excision repair. And then if we are talking about already replication errors, DNA replication errors, which is already part of the genetic information, then there is a methyl-directed mismatch repair mechanism that can be used in order to repair these errors. Okay, let's look at particular case and the sequence of actions that's happening in order to remove or to repair damaged bases. And it's important to note that we are talking about bases and not entire DNA or fragment of DNA. So let's say we have a damaged base. And what happens in cells in general, there are different proteins that are constantly scanning the DNA sequences, and glycosylases is one of them. So what it does, it scans the DNA, and wherever it sees a damaged base, particularly uracil, then it will cleave that site, it will cut the phosphodiester bond, and will leave an AP site, which is a site that is lacking purine or pyrimidine. So what happens next, then ABAP endonuclease comes and cuts the backbone to make a nick at the AP site. After that, the DNA exonuclease is coming, and it is cutting a whole fragment from that point where the cleave was made. And it cuts a fragment of DNA, and then from that point on, the DNA polymerase kicks in, and it fills in the missing complementary pairs. So after that point on, DNA ligation seals the sites of the DNA, and we have the damage repair. So particularly this is this mechanism is designed for removing uracil that was created by cytosine deamination. We talked about it previously. Again, another mechanism that we have talked about was the UV radiation, the environmental effect that was causing mutations. And also, I have mentioned that it can also trigger a repair mechanism within cells. So what happens when exposure of UV light is taking place, the complex called UVRA, UVRB is being activated, which scans for distortions on a DNA double helix. It looks for basically thymine dimers. When it recognizes it, then UVRB, UVRC complex, which is also being triggered due to UV radiation, gets activated, and it comes and nicks the damaged DNA. From the damaged part, it nicks it 4 nucleotide on one side away and 7 nucleotide another side away, and it just like removes that portion of DNA sequence, and then same way as we discussed in previous example, DNA polymerase fills in the complementary, the appropriate complementary basis, and then the DNA ligase does its sealing job. We also very briefly talked about methylation, methyl-directed mismatch. And now let's see how is this type of mismatch or mutation being repaired. So in bacteria, the methyl group is being added to adenine in GATC sequence, which is the old DNA. So that's the way that old DNA is being differentiated from the newly synthesized DNA. So what happens, mut-S and mut-L, those are the proteins that are going to help us to repair the mistakes. They bind to the mismatched nucleotides, and they cut everything between the methylated side and the mismatch, and they remove the entire thing, and then using the old template of the DNA, the new DNA is being having a new complementary strand synthesized using the old DNA as a template. Here you can see graphically what I was talking about, that the mismatched part between the methylated site and the mutation was removed, and then the new sequence was synthesized based on the the old DNA sequence. Not all of the repair mechanisms are perfect and are going to fix all the mutations and all the problems that are occurring in the DNA. However, some of them are such that it's like better than nothing, at least they can do something. So one of such mechanisms is SOS system in bacteria. So when polymerase that is used at a replication fork, it moves and it gets to the point where it's stuck and it cannot move anymore. So at that point, what happens, that fragment is being removed, and instead of that, random nucleotides are being added just to be able for the DNA to still elongate and continue its sequence. So definitely there is going to be this portion of sequence that is completely nonsense in terms of genetic information. However, it is giving a chance for rest of the DNA to keep its information intact and pass it on. Again, this is not a perfect repair mechanism. However, it's better than nothing type of mechanism. Another of those type of mechanisms is non-homologous end joining, where and it deals with double-strand DNA breaks that are being caused by X-ray radiation or reactive oxygen. And we will talk about this on our next slide with more figurative way. So here is the graphical representation of what we have been talking about. So here is a double strand that that is unrepaired. And it has breaks, which can lead to lethality. And those type of damages are due to deletions, inversions, translocations, and about all of those we have talked about during one of our previous classes. So what happens here, proteins come and they clean these broken sites, and they bring two fragments of DNA together, the first fragment and then the continuation after the broken part. They bring them together and then they just join them. Of course, some information of DNA is definitely being lost. However, it's better than not having anything at all and having a lethal outcome. So this is again one of the mechanisms of not perfect repair mechanisms. So this is great that our organism, our cells, have some mechanisms to deal with mutations. However, it's important to note and it's important to remember that all of these mechanisms of repairs are done through enzymes, are done through proteins themselves. And how are the proteins made? They are also encoded by genes, which are also potentially are able to get mutations themselves. So what happens if the repair protein, repair enzyme has mutation itself? So what happens in that case, that it will not function properly and repair the mutations. And therefore, for example, here, the disorder called xeroderma pigmentosum, which I'm pretty sure a lot of people are familiar with. It's it's due to a mutation in one of 7 genes encoding enzymes that is involved in excision repair process. And here is a picture where the UV radiation caused repair was not functioning, and therefore it is causing skin cancer excessive in its excessive form. Another example, and the picture is not definitely here, is the hereditary form of colorectal cancer, where mutation in human homologue of bacterial genes, so we talked about the mut-S and mut-L, which is involved in mismatch repair mechanism. It's mutated, therefore the repair mechanism is not functioning and the mutation is not being repaired. So we talked about those repair mechanisms. Next, we are going to talk about impact of unrepaired mutations in more depth. So what are the impacts of unpaired mutations? So mutation can be in a germline or it can be a somatic mutation. So germline mutations, they occur in gametes or in gamete precursor cells. They are transmitted from generation to generation. They are encoded in genetic information. And also they provide a source for natural selection because if the mutation is such that the offspring will not survive, then that mutation will not carry over to the next generations, obviously. However, if it's something that will make the offspring stronger, then it will help help and evaluate the species further. Another type of mutation, the somatic mutations, they occur in non-germ cells. Therefore, they are not being transmitted from generation to generation. They are not encoded within the genome. Of course, they will affect the survival of the individual. They can either kill individual directly, or they can cause in different disorders that will have a lethal effect, and such as cancer, for example. This is it for today. Next time we will talk about experiments that are worth to go over, experiments that shed some light to mutagenesis science and studies, and also carcinogenesis in general.